Okay, now to continue with this. If one parent has the allele uppercase I superscript A, uppercase I superscript B, what blood type are they going to have? They would have AB blood type. Let's say another parent has this same blood type. So it's heterozygous for A and B alleles. This is what their offspring would look like. So if my original question was, I'm blood type B, what would my chances of having blood type B be if my mother had AB blood type and my father had AB blood type? Well, the chances of having B blood type, as you can see, are a 25% chance. 25% chance of B blood type. Okay, and then what would your chance be of having AB blood type? It would be a 50% chance. And how about of type A blood type? It's a 25% chance. Okay, and if we were going to write out the genotypic ratio, what would it look like? 1 to 2 to 1. Okay. How about if one of the parents had type A blood type, but their blood type A genotype is the allele for blood type A plus the recessive lowercase I. And let's say that the other parent's blood type was heterozygous for the AB alleles. Okay, let's take a look at what that would look like. Okay. So what percent chance is there of having blood type B? Again, it's 25% chance. However, the genotype is going to look different. The genotype is the uppercase I superscript B and lowercase I versus over here where we have uppercase I superscript B, uppercase I superscript B. Feel free to rewind this video and to pause it before I complete the Punnett squares and try for yourself to complete the Punnett squares and write the genotypic and phenotypic ratios. Okay, so the next concept that we're going to get into is about what we call test crosses. These are crosses that are used with a homozygous recessive individual and an unknown individual. When we say crosses, we're talking about we are breeding a male and a female organism, and we say crossing them, crossing their genes with each other to create some offspring. So what a test cross is it's used to determine the genotype of an unknown individual. For example, here we have an unknown parent, this red flower, the female red flower. We're crossing it with a homozygous recessive known genotype of the male. Now, how do we know this male's genotype? Because we've observed data from the past to show us that the lowercase r is the allele for white flowers. And so if we see a white flower, we actually know for sure that it must have a homozygous recessive genotype, just based off past data of breeding the flowers. But when we see a red flower, we're not exactly sure what its genotype is. Why? If you said we don't know what its genotype is because we don't know if it's heterozygous or we don't know if it would be homozygous dominant, you would be correct. It could either be big R or big R little r, so either homozygous dominant or heterozygous. We don't know. So we're going to perform a test cross to know what the genotype of the unknown parent actually is. So what we do is take a Punnett square, like the ones below. We take the unknown parent and cross it with the homozygous recessive parent. And what we do is we observe the results that we see in their offspring in order to determine the genotype of the parent. So if this parent is big R and not big R small r, if the unknown is big R, do the Punnett square for that now and determine what the offspring should look like. Let's say that this Punnett square over here is the unknown parent big R small r. And so cross that with the homozygous recessive known genotype and see what that Punnett square would look like. You can pause the video now to complete those Punnett squares. Okay, and so when we did these crosses, the unknown parent number 1 and then unknown parent number 2 with the homozygous recessive other parent, we ended up with all red offspring in one of them. Okay, so what does that tell us? It tells us that this parent must have been big R, this parent. But this parent over here produced half of the offspring that are R and half of the offspring that are white. So what does this tell us about the unknown parent's genotype? It tells us that it was big R, small r, because it did produce half of the offspring as white. So this is called a test cross. And again, it can be used if you're unsure of the genotype of one of the parents and you are able to breed it with a homozygous recessive parent for that same trait that you're trying to determine the genotype of in the unknown parent. So much of this subtopic actually has to do with diseases, genetic diseases, which makes sense because genetic diseases are passed down genetically, and many of them can be determined using a simple Punnett square. So most genetic diseases are considered very rare, and so the probability of actually having a genetic disease is low. And the reason for that is that most genetic diseases would be carried by a recessive allele. That's how they continue to be passed on, through these recessive alleles that are carried by parents. Remember before we were talking about carriers? Now this is going to become more important with genetic diseases. So the probability that both parents would have the recessive mutation on the allele or on the same gene is very small, and this is why it's considered rare to actually have a genetic disease. So cystic fibrosis is one example of a genetic disease that is autosomal recessive, is what we call it. It's called autosomal recessive because it happens in your body cells, your autosomal cells, and it's determined by recessive alleles on your genes. So what cystic fibrosis does is, it is a mutation on the gene that causes this, and it basically causes your mucus, sweat, and digestive juices to become very thick. Instead of to be more lubricating and thin and helpful for your body, they become very thick, which can cause many complications. So here's a Punnett square that we have with an example with cystic fibrosis. Again, it is a recessively determined trait. It's a recessive trait. So the mutated allele, which causes this disorder, is lowercase, right? A small t is what we're going to represent it with. And so if you would just go ahead, pause the video, and try to complete this Punnett square and see what is the likelihood that these offspring of these parents would have cystic fibrosis. If you completed that Punnett square, you would see that 25% of the offspring would have big T genotype, inheriting both of the normal alleles in their genotype. 50% of the offspring would actually be what we call, remember the word for it? Carriers. Just like both of these parents are considered carriers because they're heterozygous for the trait, and it's a recessively controlled trait, right? It means that they carry the recessive mutated disease-causing allele, but they don't express it. Why? Why do they not express it? It's because they also have that dominant allele, the big T, right? That masks it or covers it up. Okay, and so that would be 50% of the offspring that would have that, and 25% chance of the offspring having cystic fibrosis. The phenotypic ratio, as you can see, is 3 to 1. Okay, the genotypic ratio would be 1 to 2 to 1. I am going to stick into these last 3 minutes of the video a little bit about pedigree charts. You will see more of this in later videos, but I wanted you to see it first. So what these pedigree charts are all about is they're a way to represent the family histories of generations of families. So the generations are noted here on the left. This is the first generation, and that would be the first generation couple, male and female right there. Females, as you can see, are denoted with a circle. Males are denoted with the square. Okay, and then this would be the second generation right here. So all of them in this line would be the second generation. So for instance, this could be the great grandparents. These could be just the grandparents, right? And then these maybe are the parents, okay? And so this is the third generation that goes along the line here. You can take a look at the key to find out more of what each of these means. If the square or the circle is filled in, what does that tell you? It tells you that they're actually affected by the disease, okay? If they're not affected, the circle and the square are not filled in, not colored in. If they're deceased, there's going to be a cross through the circle or the square. Go ahead, as an easy practice, just fill in this chart. Pause the video now. Fill in this chart with the genotypes of each of these as you move along. So what would the genotype of A, the male, and B, the female, be for this one? And give your reasoning, and so on. Now, before this video ends, I want you to practice answering a pedigree chart question. These can be slightly tricky, but it all lies in trying to determine the genotypes of each person along the way. Okay, so you have to look at the genotypes of each and try to figure them out. Yes, you can only tell if they're affected or not affected, but you are also able to see their offspring and therefore able to determine a little bit more about what their possible genotypes would be. So A and B, for instance, are not affected, as you can see in this key. But what would their genotypes have to be in order to have one child that would be affected in C right over here? To be able to determine that they are both in fact carriers. Now please answer this question, pause the video and answer this question. Calculate the chances of the child having cystic fibrosis, the child of D and money sign. If you said there was a 50% chance of the child having CF, you would be correct.